Well, I'd like to welcome everyone to our February 6th meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics in GLIMPRINT. We have a great seminar lined up for you today from Madam Knapp from University of Florida. As always, I need to remind people that this meeting is live streamed on YouTube and recorded, and an edited version of it is made available on YouTube as an open resource. As always, Tom Helikar and myself, Jim Sluka, and Lorenzo are available for your comments, suggestions, complaints. Please feel free to reach out to us anytime for ideas about how to do things better or ideas for new projects. We have our usual communication channels. They work better if you use them. We have a MSM viral hashtag. We have a GLIMPRINT hashtag. We have the long-standing IMAG wiki page. We have a LinkedIn page. We have the YouTube channel. And we'd love to have your help developing these resources and distributing them. Again, we have that Twitter handle, MSM Viral, and also GLIMPRINT. If you tag things to us, that's helpful in distribution. If you have a publication that you would like to have put up on the IMAG MSM page or on the GLIMPRINT page, please send it on to Jim Sluka. We always want to publicize new material in the area of digital twins and technology, multi-scale modeling. Please, anybody who has material that they would like to put up there and advertise, do so. We're happy to do that. The GLIMPRINT site is back up. You can put material there. Please help us build that site as well. We're very eager to develop that further. If you're willing to help Jim Sluka out in that, we would appreciate it very much. Coming up, there is the International Summer Institute on Network Physiology that might be of interest to people. End of July, beginning of August. Application deadline is March 15th. There is, I guess it's expired now, but maybe there's a late application for the Cold Spring Harbor Systems Immunology course that might be of interest to people. That's actually a place we might think about hosting workshops in the future as well. They do a great job hosting workshops, and they do take suggestions for workshops. It's an area that they have a lot of areas of interest that might overlap with ours. There is that ongoing NIH NOSI on systems modeling infection and immunity across scales that people may be interested in. Coming up, we have a speaker booked for February 20th, and Chase will come back and talk to us on March 6th. We now have a speaker lined up for April 3rd. That's not here on the books yet. If somebody would like to speak on March 20th, please feel free. As always, you've heard me say it many times, suggest yourself, suggest your postdocs, suggest your graduate students, suggest anybody who's written a paper, put up a preprint that you find interesting, and our team, Tom Helikar and Lorenzo and I, will do our best to get the people that you want to see speak up on the lineup. Don't be shy if you've given a talk for the series before. If you have new results on work you've presented previously or work that you haven't presented here previously, we're always glad to hear it. And we're always open to having experimentalists, industry, medical. Doesn't have to be a modeling talk. Anything of interest to this community is fair game for speaking. We will have a steering committee meeting tomorrow at 11 a.m. Tom and Julie have been working hard on pulling together the mini symposia proposal for SMB. They've reached out to the community at large. If you would like to give a IDT related talk as part of those mini symposia, you could text or message Tom or Julie today. They're trying to finalize that and get that set up. I think it'll be great. I think we'll have probably 2 sessions proposed for SMB, which would be wonderful as a way of publicizing the GLIMPRINT effort here. We also have been talking about how to coordinate with Anna Nirakis's huge effort on the RDA IDT project, and there's going to be some discussion tomorrow about that as well. If you're a member of the steering committee, you've already been invited. Please come. If you think you should be a member of the steering committee and you want to be, and you're not, drop a message to Lorenzo or to Tom, and we'll make sure to invite you to that meeting tomorrow and ongoing. So I don't want to take any more time away from our speaker. Today we are going to have a talk on a critical question, which is a data assimilation question. How do you use real-world data to improve medical digital twins? And I will turn the floor over to Adam for the talk.
I am going to talk about some work that I, together with Danielle Cruz, or Merritt and Reinhardt, have been doing on data assimilation for the kinds of agent-based models that often show up in models of the immune system. So I'm assuming a lot of you have seen this graphic from the National Academies of Science before. It's supposed to be a definitional thing for digital twins. It's telling us something about the way in which a digital twin is supposed to work. There's a physical counterpart and a virtual representation, and there's this flow of information between them. So physical to virtual and the virtual to physical. So the part that I'm focusing on in this is the part where you go from the physical to virtual. So how are we going to try and integrate some kind of measurements that are taken on a patient and make them part of our simulations so that they're actually, they're calibrated to the patient's current state. Maybe we learn something about some parameters of the patient and, like I said, and the actual state. So data assimilation is basically this process. There you've got observations. These are typically incomplete. What we're going to be getting out of patients isn't going to be a complete map of, where every single cell is and, what kind of state they're in. We're not going to get a complete graph of, the distributions of some cytokine or anything like that. We're going to get, proxies of these kind of measurements. You're going to get a blood panel of cytokines or, some other kinds of measurements from the patients they're definitely incomplete. And what's more, they will tend to be noisy. So there will be measurement errors. So this can be, I'm not going to worry too much in this talk about measurement errors that are systematic. We're just going to worry about ourselves about measurement errors that are noise-based. So there's also, predictions from our models. They're also sort of incomplete. Our models can't possibly capture everything. And they're going to be noisy as well. Partially, this is going to be noise because these models are often themselves stochastic. Some of the noise could be more systematic, where we're failing to capture some aspect of biology. So data assimilation shows up in a lot of engineering fields. Anything that really comes into contact with both models and actual data has some kind of data assimilation tradition in it. The particular kind of data assimilation I'm going to talk about, it has a history which partially comes from position tracking. We're going to be talking about the Kalman filter. It was used, for instance, in the Apollo guidance computer. Some more modern applications it's commonly used in numerical weather prediction. Um, so what kind of things do we actually need for data assimilation in a medical digital twin? So as I've said, we're going to need this thing to operate in a data poor environment. So you still need to be able to make predictions while the patient is, say, early in treatment. You can't wait until there is enough data to truly fit your model, because that is past the time when treatment would have actually been useful. We will need to be flexible about the sort of input data that you can acquire. Some of this will, you may get it one time a blood panel. You might get some data from, electrocardiogram. You might just get the patient's temperature or their weight. We're going to have a lot of highly heterogeneous input data. And what's more, we can't really make too many assumptions on the timing of this, because the timing of the data, it would be nice to have it regular, but in a clinical setting, it's going to depend on when the nurses show up and actually take some reading, when the lab gets back with the data, a whole number of things. Another thing that's going to be highly desirable is going to be some kind of uncertainty quantification for your predictions. As we've established, the patient, our model of the patient's state is going to be ambiguous at best, right? And so our predictions are going to be ambiguous. And so we need to be able to say, there is such and such probability that you're going to be in this range. We can't make a lot of hard and fast calls in these things. One other thing that we need, which is less obvious, is that this entire system will need to operate faster than real time. So it means that not only do our models need to be able to execute quickly, but our inference models will need to be able to execute quickly. We'll need to be able to be relatively parsimonious about the number of simulations that you have to run to get a good fit and things like that. So some of these, in particular, the uncertainty quantification suggests that we might want to look at a probabilistic model. So one thing we could use is a Markov model for state updates. The state at time t should be a probability distribution which only depends on the previous state at time t minus 1. We're going to have uncertainty in our observations. So our observations are naturally a function of the state. But since we have uncertainty, they're going to be a probabilistic function of the state. So we've also got a probability distribution for that. And this suggests that maybe what we want to do is we want to use something like the Bayes rule to, say, well, what is the probability of our state conditional on our observations and our previous estimates of the state? So Bayes' rule can be used to give you something like this. So one problem, of course, here is that the full Bayesian apparatus is slow in general. Like you have to do a lot of simulations, and this sort of violates our desire to have this thing operate faster than real time, just simply based on the number of simulations that you would have to do. There are ways of doing Bayes relatively quickly, but it's still a comparatively slow thing. So a somewhat restricted version of all of this is the Kalman filter. And it's restricted in the sense that what you do is, to derive the Kalman filter, is you assume that all your distributions are Gaussian and that all your models are linear. And this simplifies things greatly. So it's fairly obvious, right, that our models aren't going to typically be linear, and the distributions aren't going to be Gaussian. Nonetheless, the Kalman filter still is an optimal estimation algorithm for the mean squared error, right? So that means we aren't really depending all that strongly on these Gaussian and linear model assumptions. They are certainly there to derive the equations, but it's still pretty weak. A nice thing about the Kalman filter is that it's just like in the Bayesian equations there, you have a quite flexible observation model. So the only thing that you really need to make the Kalman filter work is that you need the observables to be linear functions of your state. Okay, and even there, if we can linearize, we can sort of fudge that a little bit too. It's still okay. So some other things that are nice about the Kalman filter, it has a number of variants to deal with these sort of non-linearities. One of them is this extended Kalman filter in which you do, this is useful for systems that have like a linearization because you just do the linear approximation. The one that we're going to take a look at is the Ensemble Kalman filter, where what we do is we actually have an ensemble of runs of our model, an ensemble of states, and we use that instead of directly using distributions. There are some further optimizations of this, like an unscented Kalman filter, and there are sort of square root versions of the Kalman filter as well, which I'm not going to touch on them here, but there are a lot of variations of the Kalman filter out there to deal with a number of different situations you might find yourself in. So this is this is the Wikipedia image that sort of outlines the general way in which this works. What you have, everyone can see my cursor, right? Okay. You have some sort of prior knowledge of the state. This is still going to be treated as like a Gaussian prior here. And the Gaussian prior, it's got a mean, it's got a covariance matrix. You run the model to get a prediction. Then at that point, you make some sort of observation. And the observation, again, it's centered at yk. It's got some uncertainty. There is an update step in which you get a new mean and also a new covariance matrix. And this then becomes the prior for the next step, which you then just continually cycle through this thing. Okay. So basically, it is a prediction correction algorithm. And in some sense, you want to think about this update step as it's trying to resolve the two different things that you have right here. You've got an observation. You don't really expect that this state right here, this predicted state, will exactly correspond to the measurement there. But the update step, it resolves that uncertainty. And it does it in a way that not only does it set the state to have the measured to correspond to the measurement, but it also uses the covariance matrix. It doesn't just look at the one variable that might correspond to that measurement, but it also changes the other ones with respect to the correlations between them. So you don't just learn a single component of this state vector, but you actually learn a little bit about all of them. any of the ones that are related through covariance anyway. So like I said, this thing is derived by assuming things are linear and Gaussian, and that leads to a bunch of matrix formulae for the Kalman filter. And that turns the Kalman filter, this data assimilation task, into linear algebra, which is, we know linear algebra is about as fast as you're going to hopefully get. So I don't expect anyone to look at these matrix equations right here and just sort of nod along. I just want to point out a few key features. So here we've got the Kalman gain, and it is built up of a couple of different things. P there is the covariance matrix, which shows up in a couple of places. H is the observation model. So this is the linear model, which goes from states to observations. And R here is a matrix which encodes the error, the noise in observations. So this gives you the Kalman gain matrix, which is then used to update the mean. You have your previous mean, you have the Kalman gain. This term right here is the difference between the actual observation you made and what you would have predicted from your previous estimate. So that is the Kalman gain goes from your error in observation vector space to the state vector space. So there's one other thing that I want to point out here. That's this Q. It's a process noise. This in the linear version of this is just something you can set. In applications, you would probably want to figure out how much noise the process has yourself. It is important to notice, though, that this does not show up in the inference, this process noise. So we don't actually have to worry about that if we've got a nonlinear model, because it will be sort of cooked into our estimates of the covariance matrix in other ways. Okay. So here's one thing that is pretty important about this, that it's that the Kalman filter is a state space method, meaning that the state of the system is a vector in a fixed vector space. So we will see that this thing gives us a little bit of trouble when we try to generalize this to ABMs. Okay, so I'm assuming a number of you are familiar with ABMs as well. On the right-hand side, I don't think Gary and Chase are here. I didn't see them earlier. But this is a re-implementation of one of their models that I did. I re-implemented it in Python, so it would be a little bit easier to work with our other parts of our system. But it should be fairly similar to what their output actually is. But the key thing about an agent-based model is that you have a number of agents. In each type of agent, you have the same computational model, but each one of them carries around its own distinct instance of some number of data fields. So in the kind of model that we're looking at right here, the way it's implemented is that each one of those agents there, it basically, there's a big array, and each one of them, agent 1 will be array index 0, agent 2 will be array index 1, and so forth inside of this big array. So they carry this label around with them, being where they sit inside of the array, but that label doesn't really mean anything biologically, right? It doesn't mean like your array index doesn't mean anything. So that's sort of an issue for when we want to sit down and compare two different overall states of this system, because it means that you can't just like take the data structures that we have and subtract them as vectors and just be done with it. You could maybe, if you like, try and do some sort of shuffling kind of thing to compare two instances, try and find a minimal distance between them. Which itself is sort of a problem because, now try and compare three. It doesn't work quite so well. But there's actually a deeper problem right here, and that is that in most of these kind of simulations, you can see, for instance, this happens here. There were a bunch of these little white circle agents. They are appearing, they are disappearing. The number of them is not constant. And so the number of quantities, the dimension of the space of states here that you need to describe the simulation, it's changing. So we don't have a vector space of a constant dimension. We have this thing which is constantly changing. So what we're going to do is we're going to sort of work by an analogy with, thermodynamics, statistical mechanics, where we're going to talk about microstates, which are the fully described, they're the fully described state of the system. And from there, you're going to get macrostates, what is a macrostate? A macrostate is going to be something like, the total number of cells. In this simulation, there are cytokines floating about in a spatial distribution. It's going to be like the average value of or total number of those cytokines. So macrostates are going to be these summary measurements over the whole simulated region. Microstates are going to be the fully described state of the system. The benefit to taking a look at this, in our case, is that the macrostates are going to naturally sit inside of a fixed vector space. So we will be able to perform the kind of analysis that you do with the Kalman filter on the macrostates, whereas we can't do it directly on the microstates. So this is a figure from our paper in which we sort of like, we describe this. So we've got like a cartoon model of one of these ABMs here. So we've got these, at each grid point there, you could either be blue or white, and we've got a magenta circle and a yellow triangle agent, which are floating about. So we've got here a small ensemble of microstates. And from each one of those small ensembles, you can summarize it. And here we've got like the total number of count of blues, magentas, and yellows. And so, okay, so these two things, they have the same macro state here. How do we do forward predictions? Well, we have a model. We have a mechanistic model that takes a microstate and predicts things forward in time. And so you can just run it. And then anytime you have one of these microstate ensembles, you get a corresponding macrostate ensemble. Now, what we're going to do is we're going to then make some observation. So in this case, we make a partial observation where we find out that the number of triangles is in fact 6. And so what we can do is we can look to this macrostate ensemble here, and we can update it. You can see that a couple of things have changed anyway. So instead of 846, we've got 856, 854, 845. We've got some change to our macrostate ensemble. So now the challenge here is that what we need to do is we need to find some way of generating a corresponding ensemble of microstates because we don't have a model at the macrostate level. Our model exists down at the microstate level. So we need to be able to synthesize. We need a microstate synthesis algorithm that can go from these macrostates to these microstates. Okay, yeah, so we have our prediction step, we have our update step. So these things here, what we've got sketched out right here is like an update here. And this seems like a fairly reasonable update, right? We could take this microstate. It had been at the 846. We see that we need to update it so that there's 5 of these magenta circles. And so we can just go in and we can place a magenta circle in there, right? And we can update it, and this looks fairly reasonable. In general, like you don't want to choose things. You have to be careful about this choice, right? Because if I look at the update, there's actually a whole bunch of things that are compatible here with that macrostate. Some of them could be fairly typical states, like the one that we chose. It could be maybe a little bit less typical. This one has some regularity that we might not have really expected. Or we could have one that just looks quite strange and is sort of like overly regular, like this one, you know? So we want some kind of microstate synthesis algorithm in which the microstates that we synthesize are actually going to be realistic. They're going to be fairly probable. We don't want to choose a lot of oddballs. So we worked on this with using several different models. One of the simplest ones that we used was a wolf sheep predation model, the one in NetLogo. I translated it to Python again so that we could interface with it a little easier. But with the wolf sheep predation model, it's actually the states are actually quite simple. Wolves and sheep are basically moving around quite randomly. And so if you find out that find out that you have too many sheep, it's okay. You can just like delete a random selection of sheep from the simulation, and that's about right. And if you find out that you had too few sheep, well, that can be done as well. I mean, the sheep are basically, if you look at the distribution of sheep, it's pretty much uniform and their directions are pretty much random. It acts a lot like how this really would act in the physics setting. The sheep are acting quite a bit like a gas, right? They're sort of particles bouncing around. So we tried this out because this one was relatively straightforward. We made some observations in this. We observed the count of the number of wolves, and you can see in this graph right here, we have we chose one run of this simulation as to be a sort of virtual patient. It's giving us the true value. And then we ran an Ensemble Kalman filter for ABMs using this. And you can see that you can see visually quite well where where the Kalman filter was correcting things. So you can see like right here, we measured the number of wolves and the predictions went bang right up to the number of wolves. And this happens repeatedly. And also in this, the prediction, there is a sort of growing cone of uncertainty around each one of these. And so we can see that there's quite a bit of uncertainty about where things are. But the continual way in which we bring us back up to the true predicted value really helps out a lot. And it actually, you can see it's also doing a pretty good job for grass. Grass in this case is kept relatively close to the true values. It is corrected every once in a while. So like here, go down, and the value of grass is kept relatively close to its true value. It's certainly within this plus or minus 1 standard deviation cone of uncertainty. This didn't do a particularly good job of keeping the number of sheep at a meaningful value, although at least not in the mean sense. If you look, it is certainly inside of the cone. So through all of this, I've mentioned this macrostate, but and we oftentimes think of it as being like state variables, but you can also append this macrostate. You can append to it, a bunch of parameters. You can treat parameters in this as state variables, which simply don't move. They don't contain any dynamics. Or you can treat them as state variables where their dynamics are sort of a neutral drift. And so these ones we used used a neutral drift model to on the parameters to try and estimate what they are. So it's okay. I think on estimating parameters here. I think one thing, I have a slide in my extras bin at the end there where I can show you this model in particular. You can see the wolves really, really wanted to die out. And so I think that's actually influencing a lot of what's going on here. So this returning to this model of Chase and Gare is it's the other one that we took a look at. It's a model. This is their comparative biology immune ABM. So they were taking a look at comparing the immune responses of humans and bats. We restricted ourselves to the case where it's human biology, and we're using this as a test bed to try and do our data assimilation here. But you can see some of the features of this. There's a whole bunch of moving agents. We also have a number of spatial fields. There are a number of cytokines. There's some extracellular virus. There's an epithelial layer which exists in a categorical state, which you can see as the different blocks here. There's also an endothelial layer, which sometimes you can see poking out. And so we tried this randomized thing, this completely uniformly random modifications on their ABM. It's sort of a naive first try. And you can see here's just some sort of representative fields in the macrostate. So we've got totals of several cytokines, similar things on the virus. You can see this and that. The viral ones are pretty good fits. I think this mostly comes down to the dynamics of the model, mostly being fairly similar in that range. One place where it doesn't perform too well is here in the cytokines. You can see it always wants to diverge a bit. So what we needed was a better way of getting estimates of the microstate. And so one of the things we tried was involves this technique quantization and air diffusion. So if you used a Mac in the mid 80s, you'll be somewhat familiar with this. If you've got, say, an image that is in like a high pixel, it's got a lot of colors, right? Here. If you want to discretize it, so in this case, we're thinking about like the epithelial layer. It's a bunch of discrete values. And if you want to just discretize the Mona Lisa here and saying, like pixels below a certain threshold become black, above the threshold of luminance, they become white, you get something like this, which, I mean, in this case, you can tell that it is an image, but it's not a very good thing. What you can do instead is anytime I round, say if the luminance is above the threshold and I round it up to white, what I want to do then is I introduced some error in that, and I want to push that error off into the surrounding pixels. So if I round something up to white, I'm going to want to influence the surrounding pixels that I haven't quantized to round down to black, at least somewhat, if they're. And so you get something like this. So you can also do it for multiple classes, and you can get different kind of categorical things. Okay, so how do we actually use this? Well, let's suppose that what you wanted was you wanted to get some microstate which was similar to an example microstate, but it's got more healthy cells than what you would have had before. So we want to increase the number of healthy cells. What can you do? Well, we can take this old categorical encoding. We can separate it out. They had 5 different categories for these epithelial cells. So you could, it could be that there isn't one present. It could be healthy, infected, and there are 2 death mechanisms. And so each one of these gives you a field. It's a one hot field. If you are healthy, you get a 1, and all the other ones get a 0. Let's see, these would be an apoptost one. So apoptost gets a 1 and the other ones get 0. What we can do is then just sort of scan, we can take this and encode it. And then if I wanted more healthy cells, I just multiply the distribution here by a scalar so that the amount sums up to the correct amount. Problem is this isn't the right kind of encoding. So what we can do after that is we can just run through that quantization and error diffusion algorithm there. And anytime you're too high, it'll push, like in this case, the healthy. These are all too high. They're above 1. That doesn't make sense for this encoding, but it will push it down. I want to scroll. You can see here, we're starting to acquire new healthy cells in that region. So it goes ahead and this gives us a way of using our previous ensemble of microstates to get sort of a prior of things that look like reasonable microstates. Okay, so this is another image from our paper, just showing what the difference between these are. So if the original looked like this and we wanted to modify it, you could modify it either by a completely random change so that it becomes the right kind of thing, or you could do one of these spatially aware updates. Okay, so this certainly seems like a more reasonable thing. That this is sorry, this is a much more realistic looking microstate. So when you go ahead and do this out, here's another single run. Okay, this part still is not fitting very well. But at least in the long term here, you can see it's doing quite well. In general, this does fit a little bit better than the other one. So the other thing that I wanted to mention here was that in the work that we're doing for the next paper here, we're going to take a look at AI techniques for doing this. So effectively, what we're trying to do is we're trying to sample from a distribution. It's a conditional distribution, given a macrostate, can you produce a microstate? So there are a few techniques that we're exploring. So the thing that you see here is the output of a general adversarial network, or a GAN. So I don't want to get into the details of like how a GAN is structured, but the basic idea is that a GAN is structured as a pair of neural networks. One of them is a generator, and one of them is called the discriminator. And they are set up so that they play a game against each other. So the discriminator tries to classify examples as either being, yes, that is from the data distribution, or no, that's not from the data distribution. And the generator tries to fool the discriminator. So, the generator is trying to fool the discriminator. Now, this, the way it's set up, eventually, it will get to the Nash equilibrium of the game. And the Nash equilibrium happens when the generator outputs things that are samples from the data distribution. So this is an output of one of the GANs that I trained. And this is very similar to, so these are the different components of the epithelial layer. These are components of the endothelial layer. This is it in categorical form. And this is a very realistic looking microstate for the categorical portion of this thing anyway. Yeah, this is one of the techniques we're taking a look at. Another one that we're taking a look at is based on a diffusion model. So in diffusion models, what you do is you take a look at these things called normalizing flows. And in a normalizing flow, you've got some sort of data distribution, and you just add noise to it. You add noise to it again and again and again. Eventually, you end up with just a Gaussian distribution, a pure noise distribution. And the diffusion model, what it tries to do is it tries to reverse this process. So it goes from the Gaussian distribution back to your data distribution. And the way in which it does this is to predict the added error. So what I have on the right here is an example of, this isn't the end example, but this is an example of like what this process looks like at one step. So up top, we have an original part of the, this is something that we got by running the simulation. We added noise to it, and the neural network which we trained then predicts what the noise was and subtracts it away. So it gives you back something to this. This isn't like something that's supposed to happen in a single step. What you do is you then take this partially denoised thing and you run it again to predict the noise. You keep on going back and back and back, doing this repeatedly, and you will end up with something which is a realistic looking example, something which at least, the theory gives you that it will be something from the data distribution. So this is also a fairly useful technique which we're working on. There are several techniques which you can use to guide this. You can, for instance, if I want the macrostate to be a particular kind of macrostate, I can add noise of a particular kind to get to that macrostate, and I can always arrange it so that the noise I predict will preserve that macrostate. So we can use this in several different ways to go from, we can even use it to go from examples from one macrostate to another macrostate. And this is the sort of underlying model for the stable diffusion, which is one of these modern image generation things that you will come across. It's got, if you Google it, you'll find a image of an astronaut riding a horse on the moon, which is sort of famously generated by this. So, okay. So I talked, I think it was supposed to be shorter than this, but at any rate, thank you. I could show you more, but I'll stop here because, question time and all that.
That's great. Well, I expect people will be asking some questions about details, so you could probably show what you were planning anyway.
Okay.
The floor is open to questions.
One of the things which I was thinking about when I was was the following. In ABMs, we are often concerned with spatial effects, right? So in other words, a lump of 20 cells in the center of, say, a cancer simulation is going to be very different qualitatively from the same 20 cells spread across the ABM. And one of the challenges is that macrostates don't capture this spatial stuff, which is why we don't build models on macrostates. Those are well-mixed models. We build these spatial ABMs. So I was just wondering whether, any of the techniques you talked about actually tackle the fact that that there's a spatial distribution there, which could be important depending on what we're talking about.
So the answer is, I guess, that no, but that's what I had in these extra slides. In fact, in the viral example right there, this is one of the things that we found, even in this, that there is an important spatial distribution. And that was what I was trying to do with the updates, which preserved some sort of spatial distribution here as well. And one of the things we found right here was this very strange phenomenon. When we first encountered it, we found we were trying to show that we were doing a good job. And so our measure of that we were doing a good job was surprisal, which is the negative log probability of the data, where the distribution is our predicted distribution. So you want to basically measure how surprised should I be to learn what the data was given my prior beliefs. So the Kalman filter gave us a bunch of prior beliefs about things, and we went ahead and then revealed the data and found this very strange spike. The surprisal ought to be low. Right here, it went high, and then it went low again, which was interesting, right?
Mm-hmm.
And what we discovered there with further investigation was that the trajectory is actually clustered.
Mm-hmm.
This is a PCA visualization of the trajectories, and also just looking at parameter space, the cluster. And we found out that the two clusters, the blue and the yellow, if you compare their Wasserstein distances, right where that spike happened, these two things became distinct from each other.
Mm-hmm. Mm-hmm.
And this red one, which is out here, this was also always very far from the other two, but it's diverged rapidly. But there also weren't very many simulations there, so it wasn't as easy to see in the previous slide. And these things have significantly different phenotypes. If you look at the blue is over here. One thing that you ought to care about is the long-term amount of healthy epithelium, and you can see that for the blue cluster, it goes to 0 in the long run, whereas for the yellow one, it goes to 1, or not. It about half of everything dies off, rather. And so the interpretation that we actually found for this was that this is the point where the phenotypes diverge from each other. And what was happening is that they were starting to diverge. The distribution became multimodal. And a Gaussian distribution is not a good approximation of a multimodal distribution. So everything became very strange. Everything was far away from the median, but from the mean. And the surprisal got really bad. But afterwards, since they became far enough away from each other, it resolved. Like the Kalman filter was able to decide which one of these two you actually were in. And each simulation would decide whichever one it was separately. You'd resolve to cluster 1 or cluster 2, but regardless, you'd resolve to one of them. And so one thing that we are taking a look at is trying to find more variables for the macrostate that will be predictive. And so these are typically called like latent variables. We're going to take a look at using a variational autoencoder to try and discover these, with taking a look at a few more kinds of loss functions, in particular loss functions which not just, so the way this thing usually works is what you stick in down here into the encoder, you stick in your examples, you run the encoder, you run the decoder, and you train it so that that is the identity function approximately, so that it usually can't be, because this code space here has a dimension which is fairly small compared to the dimension of the microstates. And so you can't usually do it, but you can pick out important features. And so we're going to take a look at training those important features so that they will actually be ones that might inform us as to which phenotype you're in, say, or possibly give us more insight into the dynamics of the model. In fact, the example that you mentioned there, we're writing up a grant for this right now. One of the diagrams that we're going to have is like, these two things have the same macrostate, but in those two, like these cells and these cells are completely separate, but in this, they're intermixed. That's the sort of thing that we're intending on dealing with in the future work.
In this case, you're focusing on changing the identity of, if I understand you right, you're focusing on changing the identity of the agents. You're adding or deleting agents primarily. So is that correct?
So we have a few different techniques that we have in mind. So if we go and look at, say, this, we've got agents that are all at, the epithelial cells aren't going to move around, and so they have a fixed space, and so we can change their state or maybe delete them. So in the case where you have go back. In the case where you're in ABM that's more like this, where you've got an agent that is mobile, moving around, you still, like, typically you'll want your macrostate to have something like a total count of the number of agents. So we have a few encoding techniques that we worked out for how to encode these mobile agents into a quite large dimensional space, which we then reduce and have sort of another, we use this sort of quantization kind of method to like reconstruct them again.
I guess what I'm trying to understand though is you're treating the existence or non-existence of the agent as a essentially a parameter in the simulation and their location. Was trying to think, suppose the agents had, for example, some subcellular network, some gene regulatory network or something else living inside of them.
Yeah.
And so then what one might want to do would be to change values living inside of the agent rather than deleting or creating agents. And so I'm trying to think about, I suppose stochastically I would make those changes. I would then run those simulations and see what happened from time step 1 to time step 2 in terms of matching the number of agents. And that would be how I would generate my distribution.
Well, I mean, we're taking a look at a few different things. The one that we've most looked at the moment, what you do, we're looking at ABMs where at each time step there is at most one agent of any kind in a cell. And so in that case, what you can do is you can just like, you treat this thing as a big image with a number of channels. In a true image, it would be red, green, and blue. Here, there's going to be an indicator channel that says that there is an agent of that type here. And then there are going to be a number of other channels, which are things like, for each one of the agent properties, you add a new channel. So these are going to be very sparse in general. And so what we're intending on doing is, encoding the microstate in that manner. Dimension is huge. It's a lot of data. But then there are autoencoders that you can run to try and compress this as well. And we want to take a sort of a convolutional version of this where you don't reduce the thing down to a single vector space. So this simulation domain is 51 by 51. But you can run a autoencoder to reduce the thing from a 51 by 51 times 1,000 channel image to a 51 by 51 by 10 channel image. And then the decoder there will bring you back. And then you have to do a little bit of fiddling around with this still, kind of a quantization kind of thing or a sampling in order to get the exact kind of data structure that you want. But it's that's what we're trying to do here.
I mean, it's already impressive that it works as well as it is. And so I'm not, and what I'm talking about would be basically adding one extra level. As you say, you can use the color channels basically to describe the state of each agent individually. So that makes sense. That's great. Appreciate it.
Yeah, I mean, the other sort of option here is to like try to find some really, really big vector space where this is all encoded. Unfortunately, like the only one that I could think of to try is like the symmetric tensor algebra, and it is just way, way, way too big in order to encode all of this stuff.
Well, I've just been taking notes and thinking about how I could apply it to the kinds of ABMs we develop. So I'm looking forward to trying it out. All right. Other questions? Okay, well, I want to thank you then for today. That was really inspirational to see. And I'll look forward to trying out these methods in our work, certainly. And also seeing how the AI methods develop as well. Adam, I hope you'll come back and give us another talk when you have the AI ML methods developed more and published and in preprint, and we'll get a talk on that in detail. Thanks so
Yeah.
much.